Welcome to Chapter 8, Microbial Genetics, Part 2. In this section, we're going to cover mutation, genetic transfer, and recombination, genes, and evolution. Under the topic of mutation, we're going to be talking about the types of mutations, mutagens, or these things that cause mutations, frequency of mutation, identifying mutants, and identifying chemical carcinogens. First off, let's talk about what a mutation is. A mutation is any change in the nucleotide sequence in the DNA. If there's a change in the nucleotide sequence in the mRNA or any other kind of mRNA, that doesn't count because that's a temporary change. You can always go back to the DNA and fix it. So a mutation, remember, it happens in the DNA. So let's take a look at this scenario here that we have in this figure. The cell is getting ready to divide, so it needs to make a new copy of its DNA. Well, in the process, the DNA polymerase made a mistake here. It put a T with a G. It's not a stable situation, okay, but it didn't get corrected. Now, in this cell, because there wasn't a mistake here, everything is the same and as it was previously, and then in further cell divisions, we end up with the same DNA code that we had before, and this codon, this triplet that makes a codon, still codes for cysteine. Now over here, okay, we'll say that this mistake wasn't perceived and it wasn't fixed. So in the next round of divisions, this new cell gets the previous DNA. There's no mutation. The mRNA still codes for cysteine. But in this cell line, this cell, the A gets hooked up with T, as it should be, and it codes for tyrosine. So, this is how mutations happen. This is how we get variation within the microbial population. Actually, this is how we get variation within all life. Now, the question is, if this is a mutation that does not benefit the microbe, then this particular cell is probably going to die, or it won't be able to outcompete the others that are making the previous protein. If it makes it so that it competes better, okay, then maybe this variation will take over. You just never know. Now that we've discussed what a mutation is, let's discuss the various types of mutations. Okay, in this scenario, here's the coding strand of the DNA. We've got our triplets here, our DNA words, and here's the mRNA that gets made. Here are the codons, and here is the amino acid sequence of the original DNA molecule. Now here we get a change. Okay, so this C gets accidentally changed to a T. Okay, so here the triplet reads for serine instead of glycine. There is a change in the amino acid sequence. Okay, we went from a glycine to a serine. This is a missense mutation. It's kind of like misspelling a word. Okay, I've misspelled a word. I'm still making a protein. I still have a sentence, okay, that mostly makes sense, but I've got a misspelled word in it. Now here's a situation where this T gets changed to an A and I get an early stop. So I get the first amino acid, and then from the mRNA sequence it tells the ribosome to stop early. Whenever you get an early stop, we call this a nonsense mutation. Think of it as putting a period in the sentence early. Okay, this effectively kills the gene. The gene can no longer code for a protein product, and we're going to talk about this later. Basically, the gene is no longer functioning. Now, if the microbe can survive without the gene, it continues to survive. It might not be able to outcompete its buddies, but if it's a vital gene product, say for a ribosomal protein or for a structural protein or an enzyme that it can't do without, the cell dies. Here is a situation where instead of making a mistake where we change a nucleotide sequence, okay, we call these point mutations, by the way. If we have one removed, say this A accidentally get skipped, and we go straight to the C. So we get AAC. We get a shift in the reading frame. Okay, this is called a frame shift mutation. Everything from this mutation on down codes for a different amino acid. Okay, I like to use this example as the Scantron example. If I accidentally skip a bubble or skip a question, but for the next one I still bubble in that same one, but for the question down below, everything below that is wrong, even though I got it right. Okay, this can seriously harm the microbe. It usually kills it. Sometimes they limp along. Okay, we can also have the same thing happen when the DNA polymerase stutters and adds a nucleotide. Okay, that's still a frame shift mutation. Now there is a fourth mutation that this figure didn't have, and that's where you get a change in your nucleotide sequence that does not result in a change in the amino acid sequence. So if we had a change up here that the glycine still codes for glycine, but it's using a codon, one of the many codons for glycine, but it's not the same one that it had before. We call this a silent mutation. Okay, those are the types of mutations that I want you to know and be able to recognize on a test. Now, to help you remember these different types of mutations, I'm going to give you some examples of the different types of mutations. Now, later on in the semester, we're going to be talking about types of these mutations as they relate to microbes, but we're going to talk about some human examples that you may be familiar with. Okay, silent mutations, where you have a change in the codon that does not relate to a change in the amino acids. We have done extensive research in relating who's related to whom. Okay, paternity cases. Who's the daddy? Okay, we track silent mutations. Okay, changes in the nucleotide structures that don't relate to changes in the DNA sequence, but still relate to who's related to who. Who did you get the DNA from most recently? Okay, now for missense mutations, we have some diseases that resulted from a single change in a nucleotide sequence that resulted in a single change in an amino acid that results in a disease. Cystic fibrosis is a prime example. Okay, we've got a change in one amino acid that results in mucus being produced that's thicker than being normal because of one amino acid sequence change or in the protein of the mucus. It's amazing to think of that big of a change coming from one amino acid, but it was in an important spot. And so we have too thick of mucus being produced by people who have this change in the DNA sequence. Nonsense mutations. Believe it or not, you have all sorts of nonsense mutations. They figure that 60% of your olfactory genes have been killed. They have a nonsense mutation, which is why our sense of smell is not nearly as good as the sense of smell as, say, a lab rat, even though we have approximately the same number of genes. We have in our cells these fossil genes, if you want to think about them, or vestigial genes. Okay, frame shift mutations. Usually when frame shift mutations happen in our cells, it can result in cancer, it can result in pregnancies spontaneously terminating because the fetus is non-viable. Now, like I said, we're going to relate these to microbes later on, but I wanted you to have something to hang the information on. Now let's talk about mutagens. These are agents that cause mutations to happen more frequently than they would naturally. You naturally get mutations, changes in the nucleotide sequence, just because DNA polymerase isn't perfect and makes mistakes. And so you get mistakes in the code that happen on a fairly regular basis. But mutagens cause mistakes to happen more frequently. So our first one is nitric acid, HNO2. And what happens is it changes A's so that they look like G's to DNA polymerase. It's still an A, it's still an adenine, but it looks like a G. So when we go through replication, this A on this template pairs with the C. Then as further replications go on, this TA pair turns into a CG pair. Okay. Now, here's another mutagen. These are nucleotide analogs. Here's the normal structure of an adenine nucleoside. Okay, so here's your base. Okay, 2-aminopurine nucleoside is an adenine analog. It looks just like it, there's just one small change. It doesn't have this amine group here, it has a hydrogen instead. So DNA polymerase sees this as an A, gets put in, but because of this hydrogen being here, it can form 3 hydrogen bonds just like a guanine, so it pairs with cytosine. So just like up here, we get a change of an AT sequence to a GC sequence. Lest you think that all mutagens are chemicals, let's move on to physical mutagens. In this case, ultraviolet light, UV light. What happens is UV light comes in, and particularly when you have 2 T's next to each other, this can happen in other areas, but it's especially when you have a thymine. Instead of holding hands with the A's across from them, they will hold hands with each other. We call this a thymine dimer. And it causes a bump in the nucleotide sequence. And what happens is, as the DNA polymerase comes along, this bump is too big for the active site to get around. Think of it as being a sandwich just a little bit too big for your mouth. So DNA polymerase has 2 options. It either skips the lump and you get a deletion mutation, or it can stutter at the beginning and you get an addition mutation. Either way, it's not good. So what can happen is cells have a way of repairing this. They can come in, clip out the lump, they can see that there is a bump there that shouldn't be there, cut it out, then DNA polymerase comes in and fills in the nick. This is what happens when the cell is not synthesizing DNA. If it's synthesizing DNA, or if there's too many of them, then these mutations can happen. Gets passed down to the new cell. Now let's talk about frequency of mutation. But generally speaking, we talk about mutation rate against the background. Okay, what's normally happening versus when we have higher exposure to mutation. Now there are times when in the lab we increase the mutation rate for our own purposes. When we're doing genetic studies, we will cause what we call knockout mutations, nonsense mutations in a gene, so that we can see what the gene does. When it's not there, we can see what goes missing. Okay, now DNA polymerase mistakes. Background mutation rate is generally ascribed to DNA polymerase making mistakes in adding nucleotides. Now DNA polymerase has a proofreading function, and it's very, very good at its job, and the mutation rate is very, very low. If you get a mutation in your DNA polymerase that affects its proofreading, either to improve its proofreading or to decrease its proofreading, then you get a change in the background mutation rate. Let's talk about natural selection. Those mutations that benefit the microbe, that make it better adapted to its environment, that make it so that it can outcompete its competitors, its buddies in the microbial population, then those are going to persist and they are going to spread. Mutations that make it so that it doesn't compete as well, that it's less suited to its environment, generally speaking, are going to be eliminated from the population. Now let's talk about identifying mutants. Usually in this part of the lecture I make a bad joke about the X-Men, but I will spare you that. When we are doing genetic studies, say for example, and we've just exposed our microbe to mutagen in the hopes of knocking out a gene so that we can see what happens when we lack that gene product, we want to identify those mutants so that we can study that process further. So there's 2 ways of doing it. This diagram shows what is called negative selection. So we have our microbes that have been exposed to our mutagen, and let's say that we're studying the process whereby E. coli makes its own tyrosine. So what we do is, after exposing the E. coli to the mutagens, we grow it on media that has the amino acid tyrosine. Then what we do is we take a piece of sterile black velvet, we put an X on it so we know which way is up, so to speak, and we press it against the plate. Now we pick up microbes on the black velvet. Then we press it against plates that have the tyrosine and plates that don't have the tyrosine. Then after incubating, we look for who's missing. Okay, this microbe here grew where there was tyrosine present, did not grow where there was tyrosine present. We know this is the microbe that has the gene for making its own tyrosine killed. It's a knockout for that. So we can take that off of the media that has the tyrosine, continue to grow it on tyrosine, and study that gene process. Now we also have positive selection. Suppose that we've exposed these microbes to a mutagen in the hopes that it's going to become resistant to penicillin. Then what we would do is we would take this and we would do the same thing, but we have penicillin here, no penicillin here. All of these grew. This one got killed. We're not interested in this one. We're interested in all of these. So we're positively selecting for the mutant that we're after. Before we leave mutagens, let's talk about a test that is used to identify chemical carcinogens. And it's by identifying mutagens first. Now back in the day, when we were going to be looking at as food additives, new drugs, etc. If we wanted to find out if they had carcinogenic capabilities, we would expose them to mice over long term. Now, mice generally live for about 2 years before they start getting old, so it's much faster to identify carcinogens in mice than in humans, because humans live to be 40, 50 before they start getting old and start getting cancer. 2 years is still a long time to be in testing. So Dr. Ames, of which the Ames test is named, came up with a way of identifying mutants. The theory being that if something is a mutagen, then it's more likely to be cancerous. And if we can identify that something is not a mutagen, it's not going to be carcinogenic. So we can eliminate that from testing. Okay, so here's the process. They take salmonella that is incapable of making its own histidine. It's His negative. And we expose it to whatever we're testing. If it's a drug, then we add a liver extract. Why do we do that? Because everything goes through your liver and gets changed, so we want to see what the change is, secondary metabolite, and we expose our salmonella to it. We plate it on agar that doesn't have histidine in it. Then for control, we take the His negative salmonella, put liver extract in, plate that, and just see what the background mutation to reversion to being able to make its own histidine. Okay, so the background is 2. If we see a whole lot more colonies growing, then we know that this particular chemical is a mutagen. Now, bacteria can't get cancer. Cancer is strictly a function of being a multicellular organism, where one of the cells doesn't play with the team, starts multiplying faster than it should, and ends up killing the organism. If you're a single-celled organism, you don't have to worry about that. But as I said before, if something does not cause mutations above the background rate, we don't have to go on and test it in mice. And it's cost effective and it's quicker. We can screen hundreds of chemicals every 2 days by using this method, whereas before it would take up to a year to test something using mice. And so it's quicker, it's faster, and it's also known as positive selection for your mutants. Alright, enough about mutation. Let's talk about bacterial sex. Actually, bacteria do not engage in sexual reproduction. Sexual reproduction means taking half of your genetic code and combining it with the half of a genetic code of somebody else. And bacteria don't do that. But they do take bits and pieces of their genetic code and transfer it between each other. And this process we call transformation, conjugation, and transduction. And we're going to talk about the various differences of this. Now, this type of genetic transfer is important because it leads to variation within the population of bacteria, which otherwise they wouldn't get because they're reproducing asexually. In other words, making clones of themselves. Now, one more thing that you need to know before we talk about genetic transfer and recombination are the two different types of genetic transfer. The one you're most familiar with is vertical transfer. Here we have stick man and stick woman. They see each other, they fall instantly in love, they get married, and they have a child. This is vertical transfer. This is transferring genetics from one generation to the next generation. Meanwhile, you're thinking, what other genetic transfer is there? Well, enter the weird world of bacteria. They have horizontal transfer. That's where you have two individuals of the same generation. One individual has a mutation in which he's been able to grow hair, which is unusual in the stickman world. And through the miracle of horizontal transfer, gene transfer, that mutation is transferred to an individual in the same population. Now, animals can't do horizontal gene transfer. Bacteria do it all the time. Now, when we talk about transformation, conjugation, and transduction, this is horizontal gene transfer. This is the gene transfer in which individuals of the same generation transfer just a part of their genetics to another individual. It doesn't even have to be the same species. Now, lest you think that bacteria don't engage in vertical gene transfer, they do that too. It's just asexual. Just stick man would divide himself into two and he would have two clones of himself. That would be asexual vertical gene transfer. Now let's talk about transformation. Now for transformation, it helps to have a little bit of history. Back in the day, when they were working on the vaccine for pneumonia, the main cause of pneumonia is pneumococcal staphylococcus, and the kind that causes pneumonia, the virulent kind, has a capsule. So you have, generally it's diplococci, you have a capsule. If humans, or if you inject it into a mouse, the mouse or the human dies of pneumonia. And when you pull these colonies and grow them on agar, they have smooth colonies. So they are called the S form. Well, in the process of working on the vaccine, they tried using what's called the R strain. Now, remember back when we talked about killed genes, this strain of pneumoniae does not have the genetics to produce a capsule. That gene has been killed. But the problem is when you inject it into the mouse survives, and that's a good thing. But when you injected them with the capsule-forming virulent smooth strain, then they didn't have immunity to it and they still died. So the R strain was not a good vaccine strain. So what they tried next was taking the smooth strain, the kind that has a capsule, killing it by heat, injecting it into the mouse. You can't pull any colonies from the mouse's blood, but the mouse now had immunity to bacteria that have the capsule form. That's a good vaccine strain. Well, then they tried injecting both the killed strain of the capsule form and the live strain of the avirulent strain that was not able to produce the capsule, and the mice died. And they went, huh? And when they cultured the blood, it was all S strain. So what happened? So are we talking about zombie bacteria that once they're dead, they can still come back and eat you from the inside out? No. What happened is they said that the rough strain, the strain that was not able to produce the capsule, somehow transformed into the smooth strain that was able to form the capsule. Well, they did a little bit more research, and what they found out is the bacteria were picking up DNA from the environment. DNA from the killed strain. Remember, when you heat kill bacteria, you have a tendency to disrupt proteins, including cell walls. You pop the cells, and DNA is floating in the test tube with the R strain. And the R strain is Think of it this way, picking through the recycling bin of the test tube and saying, hey, I've been needing one of those genes that actually works. I'm going to take that and I'm going to replace it with my killed gene, and now I'm going to be able to make a capsule. Okay, this is transformation. Just picking up bits of naked DNA out of the environment and inserting it into your own genome. So far as we know, multicellular organisms can't do this. But a way to remember transformation, the way that I remember it, is think of the wolf man transforming in the light of the moon. He's a normal man, but when the light of the moon hits him, he turns him into a werewolf. So think of these avirulent strains transforming into the virulent wolfman strains. I don't know if that helps, but it helps me. Okay, for this next part, conjugation, some bacteria have a plasmid. Remember back to previous chapters when we talked about parts of the cell. Plasmids are little extracellular chromosomes, circles of DNA, and some of them code for conjugation pili. And this is a conjugation pilus, and this causes the F plus or fertility plus bacterium to attach to an F minus cell, and the plasmid replicates itself, moves along the pilus, and goes to the F minus cell. This looks an awful lot like bacterial sex. In fact, that's where conjugation comes from. It's the same Latin root that we use for conjugal visits in prison. But as it turns out, it's not really bacterial sex because the whole genome is not being passed. Also, once this F minus cell picks up the F plus plasmid, it will turn into an F plus cell and it'll get all of these pili. So if this were truly sex, then whenever a woman had sex with a man, she would wake up a man. So not exactly bacterial sex. It's more like the Borg. So here comes the Borg ship, and it's the F plus bacterium, and it reels in the poor little Federation ship. Resistance is futile, and all of the inhabitants are given mechanical enhancements, and they go off and they become Borg. So I think that's a much better analogy for conjugation as opposed to sex. All that these plasmids need to pass from bacterial cell to bacterial cell is the F gene, which codes for the conjugation pili. Now, sometimes these plasmids have other genes. Some of them have R genes, which code for resistance to antibiotics. So, through the miracle of conjugation, bacteria within the environment can pass antibiotic resistance between each other. It's very annoying to those of us who are working in the clinical sciences, but it's going to happen. Okay. These plasmids can also have all sorts of other genes, but we'll stick with those for the sake of simplicity. Now for the last example of vertical transmission in bacteria, transduction. Now, transduction is transfer of genetic material from one bacteria to another one through the intermediaries of viruses, also known as phages. Now, in this SEM, you can see this poor little E. coli being attacked by a bunch of phages. So here's what happens in transduction. A phage comes down, lands on a bacterium, we'll say it's E. coli, and it injects its DNA into the bacterium. Now, with enzymes that it's brought with it, it chops up the bacterium's chromosome. That way the bacterium can't make its own proteins, it can only make viral proteins, which it proceeds to do. Makes more phage capsids. Now it also replicates the phage genome, which is inserted into the capsid. The bacterium bursts and the viruses go off to infect other bacteria. Now occasionally mistakes are made. Instead of packaging its own genome, it accidentally can package bacterial genome. Now when this virus, this phage, leaves the cell and goes on and infects another bacterium, it can't really infect it, but it can transfer the bacterial genome from the one that was previously killed to the new cell. And the new cell may say, hey, this gene is exactly what I've been needing. Okay, so phages that are capsids with bacterial genes are not infectious. They can land on other bacteriums, inject the DNA into the new cell, but it can't kill it. No phage DNA to go through the phage process. So this is transduction. Now the way I remember it is these capsids kind of look like little alien spaceships, little lunar landing modules. So the little lunar landing modules, the alien spaceships, have abducted the bacterial DNA from one cell and moved it to another cell, from one planet to another planet. I don't know if that helps you, but it helps me. Now for the last topic, genes and evolution. I've mentioned this before, but repetition is a good thing. It helps you remember. Diversity in bacterial populations are a direct result of mutation. Okay, you can't get new genes without mutation. Okay, now you can move them around within the bacterial population through transformation, conjugation, and transduction, but to come up with brand new genes, it's got to come from mutation. Now this leads to diversity. Now once you have diversity, then you can have natural selection. You can have some bacteriums that are better suited to the environment, so they do better, they divide more often, and their clones take over the population. Those that don't do as well, that aren't as well adapted to the environment, are eliminated from the population. Also, as you get diversity, maybe the new mutation is not best suited to that environment, but it's blown to a different environment where it does do better, and bacteria expand into new environments. In fact, there's not a place on the planet where you can't find bacteria. We've found them in rock samples, rock bore samples that have been taken from up to a mile below the earth. They're not happy bacteria, but they're there and they're alive. We've found them in the upper atmosphere. They're generally not dividing up there, they're just waiting to land. We've found them in Arctic snows. We've found them in salt deposits, in Andean deserts, you name it, wherever you go, you find bacteria. And we also find archaea. So because of mutation, that leads to diversity, which has led to the vast diversity of bacteria and archaea that we see in the environment today. Okay, that's it for this chapter. Once again, if you feel overwhelmed by this, repetition is the key. The more times that you expose yourself to the material, the better it is. Also, try, if you're more of a visual kinetic person, try drawing some of these things out on paper. If you're more of an auditory person, try closing your eyes and repeating what you've heard, then going back and re-listening to me, see if what you've said matches with what I say, and this will help you get the material.